ATPases in cells, and this is useful in treating heart failure and irregular heartbeat or arrhythmias. The reason why we're interested in it is that long ago in the 1980s, it was found that a subset of patients given digoxin have a high level of this inactive downstream metabolite called dihydrodigoxin. And the only difference is this double bond in the lactone ring is reduced, forming this inactive compound that no longer activates its target. If you give, it was also shown that if you look at patients on broad-spectrum antibiotics, that they have higher levels of digoxin in the blood. And even more excitingly, screens of gut bacterial isolate collection showed that only one single species does this, and that's Elenta. The problem was really nicely summarized by Ian Rowland in a book back in 1988, that although Elenta is the organism that performs this reaction, neither the presence of Elenta nor its concentration in the gut predicts digoxin reduction. And so even though this is sort of the poster child for the microbiome affecting a drug, it was not useful for anyone in medicine. And so Henry Hazer, a former postdoc in our lab, is now in industry, discovered a couple reasons why this might be. And so one is that not all Elenta strains are the same. And so we identified a 2-gene operon that we named the cardiac glycoside reductase operon. If you look at a CGR positive strain, it metabolizes digoxin, whereas CGR negative strains do not. In addition to that, CGR is controlled by the amino acid arginine, which we'll talk a lot about later in the talk. And so if you change the protein that you feed to mice that are colonized with Elenta, you can actually prevent Elenta from having an effect on the drug. And so all of this is just background to lead you up to the simple hypothesis that we had at the start of this first project, that we should be able to put CGR positive Elenta into a germ-free mouse, give it digoxin, and then we should see less digoxin in the blood because it's getting degraded or metabolized by the bacteria. And so as with a lot of things in science, the results are the opposite of what we thought would happen. This is an experiment done by Jordan Vassans, a former postdoc in the lab, who's now at Penn State. And what you can see is these are two pharmacokinetic curves. In blue, it's a germ-free mouse. And then red is the Elenta monocolonized mouse. And what you can see is that at these early time points, there's significantly more drug in circulation of the mice colonized with Elenta. Pharmacologists like to quantify these things in a variety of ways. So just to walk you through, you can look at the maximum concentration in circulation. That's higher in the colonized mice. The time it takes to reach that concentration is lower, so it's getting absorbed faster. And despite these differences, there's no change in the overall area under the curve. You can see that eventually the germ-free mice absorb the drug. But the rate is faster in the colonized mice. We went on to show that this is also true in regular mice that have a complex gut microbiota. This was an experiment led by Than Cha, an MD PhD student in the lab. And so here we repeatedly administer Elenta to conventionally raised mice. And you can see again, adding Elenta significantly increases the amount of drug in circulation relative to the control group. And that's true by these same sort of parameters. And so this sort of really led us to think about what else Elenta might be doing in the mouse that's really different from what we thought about based on our in vitro studies. And so a hint came from just reading the FDA website. So any of you, if you're interested, you can go look up your favorite drug or just guidance online. And they are obsessed with drug-drug interactions. And so they point out that drugs that induce or inhibit P-glycoprotein could potentially affect digoxin, and that this really has to be considered for any new drug that's put on the market. And so we hypothesized that maybe Elenta actually is making an inhibitor of PGP, which I'll tell you a little bit more about in the next slide, with this transporter. And so instead of a drug-drug interaction, this might be a drug-microbial metabolite interaction. Okay, so what is PGP? Many of you hopefully have heard about it, but it's a very well-studied efflux transporter. It plays a key role in two major things. One, it is important for the absorption of drugs in the gut as well as other organs. And it also is really important for cancer drug resistance. So cancer cells will upregulate the expression of PGP, allowing them to release the drug out of the cell and evade treatment. And so you can look at PGP in any cell line or in cell lines using what's called the rhodamine assay. So you add a dye called rhodamine that's a fluorescent substrate for PGP. And so if PGP is there, it'll spit it back out, and you'll have a dark cell. But if you have an inhibitor present, the rhodamine will build up inside the cell, and then it'll be brighter. And so we use this really well-established and old assay to look at a set of control compounds that are known inhibitors of PGP on the left, and then Elenta, either cell lysate or cell-free supernatant. And so what you can see is that the controls all inhibit PGP as expected. We don't really see any inhibition when we add the cell pellet, but when we add the sort of secreted factors from Elenta, we see efflux inhibition. And this is also dose dependent. If you add higher levels of media, so the media we grow Elenta in, we don't see an effect, but we do see higher and higher levels of inhibition in the cell-free supernatant. So this suggests that Elenta is making something that's a PGP inhibitor. Than went on to show that this does depend on the gene that encodes PGP. And so he used CRISPR-Cas9 to knock out ABCB1 in these cancer cell lines. And you can see on the left, the parental cell line behaves as expected. So the positive control and the cell-free supernatant inhibits PGP. But we don't see any significant differences in the knockout cell line. And you'll also note that the basal efflux is inhibited in the knockout, given that we've removed the protein. Than went on to show that this is robust across multiple cell lines and also with other substrates for PGP. It's not unique just to rhodamine. And so here, these are just 3 more cell lines. So KCO2s, HCT15, and HCT116, all intestinal cells that show inhibition in response to Elenta products. We looked at calcium AM, another fluorescent dye, and that again is inhibited. And then he looked at doxorubicin, an anti-cancer drug that also shows efflux inhibition. We also showed that adding Elenta supernatants could actually sensitize cancer cells in vitro. And so here, if we add the cell-free supernatant relative to the media, we see lower viability of a cancer cell line in the presence of doxorubicin. And so that all, suggests that, as we expected, that Elenta was doing something to PGP. And so now we wanted to know a little bit more about what the mechanism of action might be. And one of the nice things about PGP is that there's this vast literature out there, including literature on a number of small molecules that we know affect this transporter. And so each of these arrows represents a step in the PGP life that could potentially be disrupted. And so we kind of started at the beginning, just looking by qRT-PCR in these 4 different cell lines at the level of ABCB1 RNA in the presence or absence of the Elenta products. And you can see it actually goes in the opposite direction relative to what we saw with the inhibition. So we see higher levels of expression despite the fact that the activity is turned down. At the protein level, we didn't see any differences. So this is just a Western blot looking at PGP in either controls, the bacterial media, or the cell-free supernatant, and you can say they all look the same. And so this suggests that whatever Elenta is doing is downstream of translation. Next, we turn to what's called PGP Glow, which is an assay where you take the transporter in a vesicle, so there's nothing else there, and it allows a really direct readout of the activity of just that transporter itself. And so what we actually measure is ATP, which is used during the efflux process by the protein. We tested a known ATP hydrolysis inhibitor, sodium orthovanadate, which inhibits it as expected. We see accumulation of ATP, and then verapamil, which goes in the other direction as expected. And what you can see is that Elenta is also leading to this accumulation of ATP. And so that really suggests that it's having a direct effect on the transporter, preventing it from effluxing the substrate. Okay. And so all of that data was just for the type strain of Elenta. So hopefully for those of you that work in the microbiome, you're wondering, is this true for all gut bacteria or all Elentas? And so we've now tested a broad panel of bacteria. Most of these are Elenta strains, and then up here, these are sort of relatives, so other actinobacteria like Collinsella aerofaciens and others. And then this bottom cluster is sort of a group of more distantly related actinos like bifidos, and then bacteria like E. coli and other really distantly related members of the human gut microbiota. And then we ran all of these supernatants through the PGP efflux assay. And what you can see is that all of the ones at the top inhibit PGP, whereas this cluster at the bottom does not. And again, these are all Elentas and close relatives. We then looked at the genomes of these bacteria to see if there are any genes that track with the activity. And so in yellow, these are all genes that are present in all of these PGP inhibitors, inhibiting strains. And then at the bottom, if they're blue, that means the gene is absent and yellow is present. And so they sort of vary in how well they fit the pattern. And we're most excited by this group of 7 genes that perfectly fits the pattern of PGP activity. And so there's not a lot known about these 7 genes, but we sort of, stretched our annotation abilities to their limit. But we were most excited by these 2. They're predicted to be involved in the transport of molybdate, which is a really important cofactor that's used by many different enzymes in the Elenta genome. They're also, we think, interesting for future studies because they're next to each other in the genome, and then this is just some RNA-seq in vitro showing that they're expressed. And so potentially, the sort of intake of this cofactor is really important for enabling the biosynthetic pathway that leads to whatever the active compound is. And so finally, we really wanted to understand what it was exactly that Elenta is making that affects PGP. And so we used a really traditional activity-guided fractionation approach that many of you in biochemistry have probably done. And so I showed you this already. We take Elenta cultures, filter sterilize, and we found it in the extracellular fraction. We then ran it through a 3 kilodalton filter to separate large things from small things. And that data shown on the right, you can see that the less than 3 kilodalton fraction is active. But the large stuff is not. We then did a liquid-liquid extraction to separate polar from non-polar, and we found it in the aqueous or polar phase. And then finally, we ran these purified extracts on the HPLC, separating different size fractions. And you can see that fraction 3 and 4 have activity, but the others do not. And so at that point, we were feeling confident that we had really purified, one thing. But much to my chagrin, when we ran it on untargeted metabolomics, we actually found hundreds of compounds that are still in that, what we hoped was a pure fraction. And this was done with Mariah Sandy, who runs our quantitative analysis center at UCSF. We can sort of narrow down, so this is a molecular networking figure using GNPS. And they're annotated based on sort of the fragmentation patterns and retention time within each cluster of related compounds. We can then narrow it down a little bit by looking at the pattern in media controls versus cell-free supernatants. And so you can see most of the compounds we detect are actually also found in the media. And so that doesn't really fit the pattern of something that Elenta makes. And so we were most interested in these purple or red ones, the 25 compounds that are unique to the Elenta supernatant. Those sort of fall in 3 major clusters. You see these isoflavonoid cluster, carboxylic acids, and benzenes. We then did a second metabolomics experiment to try to narrow it down again. So again, we did the type strain again and then 2 additional Elentas. And we're looking for compounds that were found in the first experiment and also are present in all 3 of these strains, but not in the control. And so you can see, again, isoflavonoids are there. we see a couple of carboxylic acids, and we don't see the benzenes. And so we think that suggests that one of these two types of compounds is responsible. And then finally, we turn to control compounds that we can order from a chemical stock. And so here, verapamil is just a positive control. You can see, again, increasing levels of verapamil inhibit PGP. And these two flavonoids that we tested also inhibit the efflux transporter, but we don't see any effects of these other two groups. And so we think that suggests that Elenta is making a flavonoid-type compound that inhibits PGP efflux. And so to summarize this part of the talk, I've shown you that a single bacterial species can impact both the absorption of a drug and its metabolism, really complicating how we think about the role of the microbiome in pharmacology. We've found that our data suggests that the mechanism of action is through direct inhibition of PGP's ATPase function. And we were also surprised to see that these compounds are within the isoflavonoid family, which are typically found in plants. So these are really well studied in soy products, for example, and not something that we normally think about as coming from the gut microbiota. And so again, this really highlights the diverse ways in which the microbiome might play a role in the treatment of disease. And so all of this was sort of leading us to kind of rethink what Elenta is actually up to, and where our pet hypothesis in the lab is that it has an identity crisis and actually thinks it's a plant. So it's making these isoflavonoids, which are supposed to be coming from soy. It also has genes for making lycopene, which is the red in your tomato, and these other enzymatic pathways for breaking down drugs and other plant compounds. And so this also really highlighted the fact that, although we've studied these sort of accessory pathways for Elenta, we don't know a lot about what it's normally doing in the gut. Really, the main thing that we know comes from early work, not our work in this paper, but many decades ago, showing that Elenta requires arginine to produce energy. And it uses this arginine dihydrolase pathway to intake arginine and then make ATP. But we don't really understand what the major source of carbon is for Elenta growth, what else is required. And arginine also, I think, is never really made a lot of sense because arginine is very limiting in the GI tract. And so it seems unlikely that Elenta would have a lot of access to free arginine. And so the paper, or the project I want to tell you about today, really started by work that Jordan did with others in the lab to really collect lots of Elentas. I guess this game is back now, but Pokemon Go, we're running around collecting Elentas. And a couple years ago, he published a paper sort of providing an initial description of this strain collection that we've generated and its genomic diversity and some pilot metabolomics. But we're really limited in that initial metabolomics data set, just in terms of how many features we could identify and the data quality. And so that really motivated Cecilia, a current postdoc in the lab, to really dig deeper using more better methods. And so all of the data I'm going to show you today was done in close collaboration with Brian D. Felice and Juan Sanchez, who are two really amazing mass spectrometrists at the CZ Biohub. And they kind of gave Cecilia free rein over doing all sorts of large experiments on their mass specs. And so Cecilia really wanted to be able to better predict what conditions would allow Elenta to grow and understand what its impact on the metabolome of the gut might be. Okay. And so one of the first steps is that we had to develop a more defined media for Elenta growth. So normally we grow Elenta and other gut bacteria in what's called brain-heart infusion media, which is what it sounds like. It's brains and hearts all mashed up. And so that's great in terms of getting lots of Elenta cells, but terrible for metabolomics because you just get heart compounds back. And so Cecilia stared at the Elenta genome and came up with a list of 14 amino acids that she thought would matter or would be needed, including arginine. And then she put in some other organic acids, vitamins, minerals, and some salt. This is the final formula for her defined media, number 1. And you can see that this media actually does pretty well, so we reach ODs that are somewhat comparable to what we normally see in BHI for the type strain of Elenta. And so now that we have this defined media, we could run different time points from in vitro growth of Elenta on the untargeted metabolomics pipeline. This heat map is sort of the result from that. So you can see we're going sort of down in terms of time point. So these red features that go to blue are things that Elenta are using. And then most of this heat map is actually made up of things that are increasing in abundance during growth. So Elenta's metabolic products. You'll also note that most of these things don't have a name, consistent with a lot of metabolomics data. So we can really annotate just a small sliver of what Elenta is doing. Some of this really made sense. So you can see arginine goes down, citrulline goes up, consistent with that old arginine dihydrolase pathway. But we wanted to more directly test which of those pathways was important for growth. And so Cecilia turned to leave one out experiments where she took the defined media and made every single version of the media in the absence of one of the components. And so each of these little graphs is a given nutrient that we've dropped, and then the gray line is the control in the full media. And so we're sort of grouping by amino acids on top, and then these other nutrient components. And so, some of these we expected. So if we drop out arginine, we don't see any growth of Elenta. We also expected to see these other amino acids would matter given what is in the genome of Elenta. It looks like an oxytrope for tryptophan, for example. But we were really surprised to see this strong dependence on acetate. So acetate, as many of you may know, is called a short-chain fatty acid. It's a common end product of bacterial fermentation in the gut. And we actually don't know much about how Elenta handles acetate, but the one previous paper that's been published suggests that it actually makes it. And so this is kind of the opposite of the graph that we saw. And so we wanted to just confirm that acetate indeed does help Elenta grow. And so these are just repeat growth curves. You can see the no acetate condition, it grows a little bit, but not great. And then adding higher and higher levels of acetate gets you more Elenta growth. This effect is also really specific to acetate. So if we look at other short-chain fatty acids like butyrate or related compounds, we don't see any effect. But again, we see that acetate promotes growth. We also found using metabolomics that acetate really has a broad impact on Elenta metabolism. So all of these compounds are significantly different in the presence or absence of acetate. And you can see this sort of suite of compounds goes up in abundance, and these ones are used less in the presence of acetate. To really better understand exactly what Elenta was doing with this compound, we turned to stable isotope resolved mass spectrometry. And so here we can take a fully labeled acetate where both of the carbons have a heavy, or the heavy version. And then you can distinguish the heavy carbon from regular carbon by mass spec. And so these are just some controls to show you that if we don't add any acetate or if we add the normal unlabeled acetate, we don't see any label in the data. But if you add labeled acetate, it increases over time with growth. And then you can look both intracellularly and extracellularly and see where exactly the label goes. And so here we're just kind of counting labeled carbons. This feature, UDP N-acetylglucosamine, is really highly labeled. And then we see other varying degrees on these other compounds. And so this data obviously is very complicated, and so Cecilia has tried to summarize it in this maybe even more complicated graph. What we're showing is just these labeled carbons in green from acetate sort of moving through metabolism. And so all of these compounds where the green shows up are things where we've detected the label from that initial acetate. And so hopefully what you can take away from this is that acetate goes into all sorts of pathways that are essential for building the biomass of Elenta. And this is really in contrast to what happens with arginine. So if we do the same experiment with a fully labeled arginine and then put it into Elenta, it really just does the normal dehydrolase pathway that we knew about, with a couple exceptions related to glutamate and pyrimidines, but we really don't see that go very far into metabolism. And so that really suggests, again, that arginine is the energy source and acetate is the carbon source. Okay. And so having done all this metabolomics, Cecilia was sort of emboldened to then go back to the computer and try to build a more predictive model of Elenta's metabolism. And so she started with kind of an automated metabolic reconstruction from what's called Agora2, which is a database that Inez Thiel's lab has developed. And if you're interested, you can go and look up your favorite species in Agora2. And so, normally these are just the number of genes, reactions, and metabolites in the model. The original reconstruction is on the left. And then after all of Cecilia's hard work, you can see these bars change a tiny bit, but they matter. And then this also just shows that in our defined media, the model thinks that only a subset of the reactions are active. And so some of the reasons why we think this sort of curated model based on thinking more carefully about what we know about Elenta is better are shown here. And so here she's doing a flux balance analysis of Elenta, looking at its dependence on acetate. And you can see that the original model that we downloaded from Agora2 thinks that acetate doesn't matter at all in the blue line. But the curated model shows a dependence on acetate. And so that fits the data better. And we also see a stronger dependence on arginine in the curated model. And so now that we have a model, we can sort of play around with it. You can do anything. Maybe we're going too far off the deep end, but you can judge for yourself. But one thing you can do is try to better predict computationally what Elenta needs to grow. And so we ran the same sort of media conditions that we had already tested experimentally, and then just looked at the overlap. And so 26 nutrients that are in the defined media, both the model and the model and our experiments agree will have growth. 3 they agree will not have growth, and then you can see some of them are disagree. And so that individual overlap is just shown here. You can see simulation on the top and then experiment on the bottom. And a lot of where the model and the experiment disagree relate to minerals, and we think this is actually probably an experimental problem on our end, that we didn't fully scrub these compounds out of the media. But for the most part, they overlap in terms of the predicted oxytrophies. The next thing we did was ask whether or not this model could predict flux through different metabolic pathways, and whether or not that would relate at all to the expression of those genes during growth. And so this graph just shows you what the model thinks are secreted compounds versus compounds that are taken up in the media. And then the colors are data from metabolomics. So in red, these are compounds that go up over time by metabolomics, and in blue, compounds that go down. And that really fits the pattern of what the model predicted. Whereas this group in the middle, where the model is confused, is also confusing by mass spec. We also saw a correlation, although not perfect, with gene expression. So this is just the average basal gene expression in our media with respect to the flux through that pathway in the model. And you can see that genes that are expressed more highly are also thought to be more important by the model. Finally, something that we can't do currently experimentally is just simulate a knockout library. And so we asked, what happens if we knock out every single gene in the Elenta genome, and how would that affect growth, predicted growth? And so in the unconstrained model, or in the media that we're using, if it's blue, dark blue, that means that the model thinks it's essential, and then light blue, it thinks it has an effect but not fully essential, and then gray, no effect. And so one way to kind of validate this data is just to look at how that correlates to naturally occurring Elenta strains. And so we looked at the fraction of strains, naturally occurring Elenta strains that have a gene of interest, and then grouped them by genes that the model thinks will really affect growth versus what the model thinks won't affect growth. And so hopefully what you can see is that the kind of model predicted essential genes are much more conserved in the Elenta pangenome relative to the rest of the genes. Okay. And so next we wanted to kind of see whether or not all of this metabolism that we're seeing for the type strain was true for the rest of our strain collection. And so Cecilia took 30 different strains, mostly Elenta strains and some close relatives, and ran them through the same metabolomics analysis. And so one thing we had seen previously based on our lower resolution data was that the metabolites really group into kind of two bins. So there's compounds that are both produced and depleted that are consistent across all the strains we test. And so those are here. And then there's also a lot of compounds that are really specific to one or a small number of strains. This is just another way to look at that kind of massive amount of strain variation. And so this is a volcano plot for the type strain, just comparing the fold change of different features in the spent media versus media sterile controls. And so you can see on the upper right side of this volcano plot, all of these gray compounds are things that every Elenta strain produces, whereas the red ones are things that just the type strain, or a subset of the strains, produce. And then on the left, you can see that there's really much more consistency. So most of the things that are depleted are consistent across strains, with the exception of these green ones. We next asked whether or not we could predict this based on genes or phylogeny, and neither did particularly well. And this is just a kind of a fancy way to correlate the data. And you can see in the blue, these are the phylogeny or evolutionary relationship between strains, and red is the metabolite profiles. They don't really correspond very well. And we get a significant, but still kind of ugly relationship between gene family levels and metabolite levels. And so there's a lot of sort of unaccounted for strain variation in metabolism. And then I also, just to give you a sense for what you can now do with this sort of paired metabolomics and genomics data, we can use it to start to better identify our new metabolites of interest. So an example here is these two unidentified features, 187 and 189. It showed this really nice kind of binary pattern where some of the strains don't have that feature present and some of them do. And same for this one. Whereas ornithine, just as a control example that everything, all Elenta do or make, is really consistent. And so when we looked at these, when we compared this metabolite pattern to the genomes, we found that there are two genes, TAR I and J, that are always present when the feature is there. And these are known to be involved in making teichoic acid in gram-positive bacteria. And so the prediction is now that these are somehow related to making cell wall. And we're working to validate that now by mass spec. In addition to that, we can use this sort of data to better identify oxytrophies. So we looked at vitamin B5 or pantothenic acid. And it's a little noisy, but you can see the levels of B5 really vary between strains. And that most of the strains that are at the low end of the spectrum lack pantothenate synthase in their genome. And so we then predicted that these sort of pantothenate synthase absent strains would be more sensitive to that if we leave it out of the media. And that was true. So strains without pantothenic synthase really grow much more poorly in our defined media without the vitamin versus the other ones that are unaffected. And so finally, I want to show you a little bit of data now extending this approach to look at what Elenta is doing in the context of a host. And so we turned to germ-free or notovatic mice. We took, in this experiment, 3 different strains of Elenta used them all to colonize germ-free mice. We waited 2 weeks, and then we collected serum and intestinal contents and ran it through the mass spec. And so this is just the data at a really high level. You can see in these PCOA plots that it's pretty hard to tell the groups apart in the serum and the ileum, but you start to see separation in the cecum and colon with these sort of gray germ-free dots separate from the colonized groups. And so this, together with some of the data I'll show you in the next slide, really suggests that Elenta is having the biggest impact within the gut, and that a lot of its metabolites are not escaping into circulation. Comfortingly, we saw a lot of what we had originally seen by in vitro data was actually still true in a mouse. And so these are just scatter plots looking at the fold changes of different compounds in vitro on the x-axis versus in mice on the y-axis. And so you can see, especially in the colon and cecum, there's a lot of these green dots that represent compounds that are significantly different in both experiments. And that's true both for the type strain and these other 2 strains we looked at. But we really did not see much overlap at all in the serum or in the ileum. So really very different, even though there are things that are going up in the serum, they're not consistent between the 2 data sets. And then the last thing I want to show is that we then mined this in vivo data to get a better sense for what we think Elenta might be doing within the mouse. And so these are sort of the top compounds that either go up or down and are annotated, looking at the small and large intestine of mice. You'll note that arginine is not on here, so the one thing that we think should matter isn't on the list. But agmatine is, and we were very excited about that because agmatine, from work in other bacteria, is known to have a very similar pathway to what arginine does. And so this is just the agmatine dihydrolase pathway. It, just like arginine, it can get taken up by bacteria and then used to pump out ATP. And we kind of felt foolish that we didn't find this earlier because if you look in Enterococcus faecalis, for example, there's 1 cluster of genes for using agmatine. But if you look in the Elenta genome, there's actually 3 whole copies of this cluster. There's actually more genes for dealing with agmatine than arginine in the Elenta genome. And so we think agmatine actually provides a nice alternative to arginine in the gut. Again, this is just a summary of the pathway and the genes that are involved. We did some experiments in vitro to show that you can sort of mix and match arginine and agmatine. So if you do, for example, 50-50 arginine-agmatine in purple, is sort of similar to just having the full amount of arginine. And then we also looked by RNA-seq at gene expression and found that genes that we predict to be involved in this agmatine dihydrolase pathway are significantly upregulated in the presence of agmatine. Okay. And so hopefully I've convinced you that this sort of systems level approach that we're taking is starting to give us some insights into this really different metabolic strategy that Elenta has in the gut. We've learned that it does rely on arginine and or agmatine for energy and acetate for carbon, as well as a variety of other nutrient dependencies. And we think that this has potentially important implications now when we start to think about the role that Elenta plays in the gut, both in terms of its potential effects on the host and how competition for arginine and acetate and these other compounds may influence its effect on other bacteria. And so with that, I want to thank you guys for taking that deep dive into Elenta biology and metabolism and for having me here today. And happy to take any questions. Sure.
Flavonoid, so it's the first story. So I was wondering whether there's anything in the meat. So Elenta, as you know, is really good at transforming flavonoids. Whether there's anything in the media that, rather than being synthesizing the flavonoid, just transforming something that is in the meat.
Yeah, we were really worried about that, and we think it is not in the media. So I didn't have that on the slide, but we've also done it in the defined media, and there's the same effect. So it doesn't need anything from BHI or other medias. And we've also looked by mass spec now in multiple experiments, and we never detect these compounds in the media. But,
So in that story, when you look at the impact on the metabolome, so whether it would be productive to sort of look at the effect of the lenta in the context of sort of a community where there's acetate, even so the impact that you show in vitro acetate has and sort of what this organism does.
yeah. Yeah, that's definitely where we want to go next, is like competing it against acetate producers or other consumers, and see how much that affects, both the overall pool of acetate in the gut, but also the growth of Elenta.
It doesn't seem to change what it does, though, with acetate, right? In vitro.
It doesn't change or what do you mean?
It changes what it does.
It does, yeah, definitely. In vitro, for sure. Yeah. But yeah, we haven't done any experiments yet in mice now to like tune the level of acetate. Yeah.
I have a couple of questions. One is, I'm guessing there are multiple inhibitors that get shut down at PGP transporter. Is that correct? Or is that molecule that you had as your control the only positive control for that transporter?
Yeah, so there are both synthetic inhibitors known as well as inhibitors that are found in plants. So the two that we showed are actually they're found in potential dietary sources of PGP inhibitors.
So I guess potentially there's an entire slew of molecules that may have that same effect that the flavonoid does.
Mm-hmm.
Is that fair to say?
Yeah, it could definitely be like a combination of Elenta plus like consumption of dietary inhibitors.
Or by other bugs in the
other bacteria we haven't exhaustively tested all bacteria, but we've never seen this activity, as I showed you, like anywhere. If we go beyond the Agarthalaceae, we lose it. So at least like the isolates we've tested so far, it seems really specific to this cluster, the lineage.
I guess my second question was on the relationship between the presence of the bug and clinical outcomes. Does that mean that if you have that bug, then you're more susceptible to, say, chemotherapeutics? Is that the idea? Is that the relationship between the presence of the bug and, I guess, cancer treatment?
Yeah, we haven't done that. We haven't made it back to complete the circle for doxorubicin, or other anti-cancer drugs. We'd love to model this where you can look at drug efficacy for cancer. For digoxin, the data I showed you at the beginning suggests that this can really change the circulating levels of that drug, and that is very dangerous for digoxin. And so, one of the nice things about working in mice is that mice have a mutation in the target of digoxin, so that we don't have to worry about that in our mouse experiments. But if you're in a human and you change digoxin by that effect size I showed you, that could be really lethal. So if this all holds true in humans, it could be really important.
I have a question. Just curious about your metabolic niche that you've kind of uncovered for Elenta.
Mm-hmm.
Does that provide any insights into the sex differences? Like the fact that Elenta is like higher abundance in females versus males, and like
You're giving away my secret stuff I told you in your.
And I was just curious about the fact that the metabolites also vary depending on the sex.
Yeah, I don't know. What Ophelia is referring to is that I told her under strict confidence. Elenta is enriched in females. Yeah, I don't know if it's. We're trying to learn more about agmatine. I didn't really talk about that very much, but it's a host metabolite related to arginine. So we haven't found any literature to suggest that agmatine is sex biased, but yeah, it's an interesting question. right, for any of these compounds. Because yeah, we'd love to know if the the differences in Elenta level that we're seeing in females is driven by changes in these substrates that it needs to grow, or something more complicated. Yeah. Yeah.
This is more of a broader question, but you linked the inhibition of the ATPase site PGP to metabolites from metabolites that the link is producing. Is it broader spectrum than that specific inhibitor? Is it inhibit ATPase and beyond that too?
Yeah, that's a great question. And yeah, unfortunately, that was actually the final, part of our master plan for Than's thesis was to go there, but he just graduated. But yeah, we'd love to. Well, we've done a little bit. So we have tested other transporters that are important for cancer drugs, like BCRP, and we don't see any effect. So, it's not like it affects all transporters, but yeah, I'd love to test things that are like, for example, bacterial homologs of the of PGP, something that's closer to PGP but in a different organism, But yeah, we haven't done that yet. Yeah.
Wonderful presentation. The acetate as a carbon source is very fascinating, especially with some of the other metabolomics data you showed. So I was just curious, do you know if Elenta has any of some of the enzymes to convert butyrate to acetate potentially as a source of carbon for itself, especially even though butyrate's going to be a lot lower than acetate in the gut?
Yeah, that's actually, so I kind of yada-yada'd over that part, but that previous paper that was published suggested that Elenta is an acetogen, so it can take hydrogen and CO2. It has the genes to potentially make acetate out of hydrogen and CO2. And so one of the big things Cecilia did when she was printing the model is just get rid of that reaction because it really screws up all the predictions, But it's possible under the right condition we could sort of force it. I would love to do the kind of stuff Federico is doing in the Garden Lab and put like higher hydrogen pressure. We're just growing under normal gas levels, but maybe it can do both. It's just condition dependent. But, yeah.
About the spectrum of natural hosts for this book. Is it humans and mice? It is in your lab, I guess.
Only in our lab.
Is it all mammals, all vertebrates, all animals, all plants? Does it, colonize clover?
Yeah. I guess the short answer to that question, I can tell you all about the places I've looked for Elenta unsuccessfully. Because I really, I think that one of the mysteries is why these genes for digoxin reduction exist at all, because, mammals can't eat these things because they're so toxic. So it's an invention of modern medicine that we even consume digoxin. But the short answer is that Elenta is as far as we know, only in humans. And I think it's potentially unique in that way. A lot of other bacterial species that we see are, there's one macaque that is Elenta positive, potentially due to cross-contamination. But yeah, we've tried in our hands to get it out of non-human primates, and we can't find it. So yeah, it really does seem to really have been specific. Yeah. Yeah. I think the sex bias is fairly consistent. Africa, we don't have a lot of samples from Africa right now, but yeah, it's a really weird case where it seems very specific to humans, but also very widespread. So how that happened is an open question. I should say the relatives are more, if you go out to other, it's not like all actinobacteria. So, bifidobacterium are the sort of counterexample. Between Elentas?
Yeah, or between Elentas
Oh, yeah, I don't know about that, but we definitely see the genome. One of the reasons why we can look for these associations in the Elenta genome collection is that there's a lot of gain and loss of genes in the species. And so we do see a lot of variation in terms of genes that might be getting shuffled around in the population. We've never seen that, we've never really looked for that moving out of Elenta, you know? Yeah. Yeah. But it's a great question. Yeah.
I was just curious if you had any thoughts about like how to use genome scale models in more physiologically relevant contexts, like in the microbiome, say in a mouse. Do you have any or like have you thought about testing to see what's, Elenta's using in a mouse?
Right. Yeah, we haven't really gotten to the point where we're trying to predict in vivo growth conditions. The plan still is to kind of move our way, instead of going to the host immediately, to sort of to go towards co-cultures of other microbes with Elenta and see if we can predict it in the present, predict growth or sensitivity to other bacteria. But it's a work in progress. And potentially what we're going to need, maybe we need to start with E. coli or another organism where we already have a model that is relatively accurate.
So I guess if you have metabolite information in vivo, maybe that could be used as an input to
Mm-hmm.
genome scale model.
Yeah.
Metabolomics of the germ-free mouse, you know what metabolites are there.
Right.
It just seems like that could be very interesting if that was
Mm-hmm.
possible to predict.
Yeah. I definitely think I'm not super enthusiastic about like only pinning it on the Agora 2 models. Like I feel like it's gonna require some pruning in experiments for whatever new species we add to the mix. But yeah, other people are doing that for other, you could sort of limit yourself to species that other people are really validating some of the predictions. But,
Great, any other questions? All right, let's thank Dr. Turnbaugh for. Was that actually top secret information?
no, it's fine.